So you need to draw plan drawing of this part. Okay. Ruth, did you come to practical this week? Ruth, you came? So 5 marks for the plan drawing, and then make sure that every time after you finish drawing, you go back and read the question. Do you need to label or not? Do you need to label everything or only label one thing? Okay, every question definitely got this thing. Some students, they didn't label, they forgot to read. So always after drawing, read back the question, see that you complete the question before you move on, okay? So for plan drawing, so by now you know how to put your slide in the microscope, okay, so that you can see the correct orientation, or you also turn your paper. So this is what you see. You're supposed to draw plan drawing for this section only of the leaf. Based on the question, you need to draw this section. So if you look at this box here, there are 2 vascular bundles at the side that you need to draw, okay, other than the one in the center. Then at low power, there are certain things that are clear, certain things are not clear. So which part are not clear? This part is not clear because you do not know whether this part is one type of cells or 2 types of cells. So you need to go to high power to check, to draw. Same as for this part here. Okay, the rest are quite easy to distinguish. So you have to zoom to higher power. So this one I've zoomed to higher power and show the drawing, how to make the plan drawing. Okay. So you can see that plan drawing is just that you are trying to report exactly how the slide looks like, how the specimen looks like. So I'll expect your upper epidermis should be thicker than lower epidermis because the cells on upper epidermis is bigger than the one in lower, right? And then I need to draw the borderline between the palisade and this group of cells which are not palisade mesophyll cells. And these are spongy mesophyll cells, and this group are not. Okay, and based on theory, in the leaf, on top of vascular bundle is xylem, at the bottom is phloem, and the center is cambium. So it's thick enough for you to use 2 lines to draw the cambium. And where is the borderline between the phloem and the parenchyma cell? You look at the size of the cell. These are tiny, but suddenly this is very big. So that will tell you these 2 are different type of cells already. So you have to draw the borderline. So borderline is for you, when you draw borderline, it's based on size of cell, shape of cell, color of cell, and also your theory. So make sure the reference I've given to you for the plant slides, that reference, read everything. Once you read everything, go through one time, any slides that come to you is very easy. Okay? So you need to do your homework. Okay. So here you can see that these 2 types of cells are different. So I'm going to use a larger magnification to show you. This is even larger, so you can see very clearly. So that's why even you draw plan drawing, you need to go to high power because you need to check. Can you see this group of cell here? The wall is very thick and all are same thickness. So this is actually sclerenchyma with lignified wall because it's pink color. But this group of cell is obviously the wall is not thickened. So this is another type of cell. This is actually the parenchyma. So you need to draw one line here. And then this is another group of cells here which are not the xylem. Okay. Then from here to here is the xylem. This is cambium. Cambium, some of you don't know how to see cambium. You zoom in and see the cambium is actually a group of very flat cells like this one here. You see this tiny rectangular shaped cell, which is not, when the wall is not thickened. So this is actually the cambium cell. Can you see here? 1, 2, 3, 3 of them here. Can see or not? This cambium cell, usually about 1, 2, or 3 layer of cells. So this is cambium. This part here tear away already, so you cannot see the cell. Okay, then this is the phloem already. Okay, all the way is phloem. And then this one got chloroplast, right? This cells here got chloroplast. So this palisade with chloroplast, right? This spongy mesophyll with chloroplast, spongy mesophyll cell. Okay. Some students draw air spaces. Some students even outline the air spaces. First thing is plan drawing means you outline tissue, right? Plan drawing, you outline tissue. Tissue is made of cell, isn't it? If this one is made of cell, why do you go and outline this one? And are you trying to show that this is a tissue made of cells? The fact that this whole tissue is called spongy mesophyll. Spongy means a lot of air spaces, right? That means this tissue has a lot of air spaces. That's why it's called spongy mesophyll. Spongy means a lot of air spaces. Meso means center. Phyll means leaf. In the center of the leaf between upper and lower epidermis is all called mesophyll. The bottom is spongy, that's just called spongy mesophyll. The top is palisade. So that's how the name is given. So when you study biology, please pay attention to why this name is given. Then you can learn better. Okay, then you will not make the mistake and then start to draw the air spaces as the tissue. And also, definition of tissue is what? A group of cells. Correct? So when you learn a definition, biology is actually very easy. So you need to put the effort to learn it up. Okay, I can teach whatever, but then you need to put effort to learn it up. Okay. So this is a plan drawing. This is very good. Can you see this? Very good. 2 layers. You see not 2 different layers of cell, accurately drawn. Okay, and here, and this line separated. So this is very good. And then you see the vascular bundle got another extra layer outside there. It's actually bundle sheath. So look at the bundle sheath here. Right? Can you see? I make it bigger. Can you see this group of cells that are very big that surround the vascular bundle? And you have got so many for you to verify whether it's all of them got this or not. Okay, you see all of them got this thing here. So this is actually a layer of cell that form bundle sheath. So you need to draw this layer. And this layer of cells can be spongy mesophyll cell, and then you got chloroplast as well. But it surrounds the vascular bundle. And then the student said, teacher, I don't know how to draw the borderline between xylem and phloem. Use color to help you and use the shape of the cell to help you. So if this vascular bundle, then you can see that this is very dark. This is not so dark. So this is the line. Finish. Once you know how to draw this, you know how to draw the rest of them. It's roughly the same. You see here, this one is very obvious. This is dark. This is lighter. So the borderline is here. This is dark. This is lighter. Maybe the lighter one is cambium also. Okay, so use that one to help you. So this is for the leaf. So let's check the mark scheme. So what is the answer given for this? So please mark it. Now this mark scheme is very interesting. It says, number 1, your drawing size must achieve the minimum size and there's no shading because it's plan drawing. All biological drawing got no shading. What is the minimum size? Use your ruler to measure the space as given to you in the question paper. You see this one, this is the end of the question. And you look at the entire space, how big is the space? Okay, so use a ruler to measure from here all the way to here. At the bottom of the 5 marks, the minimum size of drawing must be half, at least half of this space given to you. So measure the size of your plan drawing. That means this is your plan drawing, right? Okay, this is your plan drawing. Minimum size. Measure the widest region here of your leaf here. Does it achieve the minimum size of this space given? So that's called minimum size. So try to draw big. Okay, so that's first marking point. Size is 1 mark. Secondly, no cells and drawn in the correct region of the leaf. No cell because it's plan drawing. Correct region means you're drawing what they ask you to draw, which is the mid-rib and a little bit of the lamina. Okay, so if you don't draw lamina, so I will not give you mark for second marking point based on the question. Number 3, shows area above the vascular bundle. Area above the vascular bundle is like this one that I have already shown you just now in the sample answer. the student draw this part here. Area above. I'll expect to see at least one. Okay, this one 2 is very accurate, at least 1. Okay. Label line and label to show the vascular bundle. So you just point to one vascular bundle and label it. Okay. So you see how the student label this one here is this labeling is wrong. I will not give mark. Do you know why is it wrong? This student's labeling is wrong. You know why or not? Yeah, he's touching this space here and say vascular bundle. This one is palisade. This is spongy mesophyll, so he's wrong. So you need to touch this whole thing. That's vascular bundle. Or you can from here to here, so can I? The big one also can, the small one also can. Okay, but you have to touch it. Vascular bundle. Next question, 2A. 2A is a very interesting question. So you learn that you need to actually read the question, then you can answer the question correctly. Okay. So you're supposed to select one large xylem vessel element and the 3 adjacent smaller cells. So I'm expect to have one large cell and 3 adjacent cells that are smaller, because it's stated here smaller, and must touch this large cell. So then they leave you to do the drawing. No, it actually says each smaller cell must touch the large xylem vessel element. So it tells you what you need to look for. And at least one of the smaller other, wow, this criteria a lot. And also it must touch at least of one of the other smaller cells. So that means to say that I must have got a big cell a big cell and 3 other smaller cells beside it. And not only that, this smaller cell must touch the next smaller cell. It must touch the next smaller cell like this. So I have to draw like this. I cannot draw individual cells separated by S spaces 1. So this is the criteria of the question. So make a large drawing of these 4 cells, only 4 cells. Use one root label line and label to identify the cell wall of only one cell. So based on this question, when you go to the slide and choose which part you want to draw, right? You go to here. Some students came to this side and try to draw from here. If you look into every single cell in the xylem here, all of them got the same size. You cannot get your answer from here. All of them. The only place you can get your answer to draw, according to what the question wants, is only this region, because this region got big cell and small cells in the xylem. So you have to come here to draw. Okay? So let's zoom in to look at this region, how the cells look like before we proceed. Can you see or not? So it's in this region that you can choose one big cell. Where's my one big cell? I can choose one big cell and 3 smaller cells at the side. I can choose one big cell and 3 smaller cells at the side. Okay, I think this is a very good one. One big cell and 3 smaller cells. And you look at how thick is the wall of this one. So that's why I've given you two drawings here for you to verify for yourself. Which one is more accurate? Okay, this is even bigger. Okay, never mind. So I've got 2 drawings here. Which one is more accurate? The one on the left or the one on the right? Based on what you can see from here. You think left is accurate or right is accurate? If you're drawing xylem vessel, you need to choose the thick one, because xylem vessel got lignified wall, not the thin one. Because in the xylem, there are 4 types of cells, right? The xylem vessel element, the sclerenchyma, the parenchyma, okay? And then what else? Sclerids. So if you're choosing the thin wall to draw and you show the thin wall, that means you're not choosing xylem vessel. You might be choosing parenchyma cells. So therefore, I will accept this as correct. This one definitely not correct really. Okay, so you check your own drawing. See the thickness of the wall. Is it representing that actual thing that you're seeing or not? Because you can see now very clearly that this is actually not correct, even though I asked the student, are you sure the wall thickness is correct? Yeah, teacher, is correct. Now I show you the picture. You tell me correct or not. Unless the student is not drawing xylem vessel, drawing parenchyma cell lah. Then wrong already also. Next thing is label cell wall. This one got no label cell wall. Drawing very, very good, but then -1 mark. You see or not? Your drawing can be accurate, but then you forgot to label, you also same mark as this student. Correct or not? This student lose mark for accuracy, but label. This student lose mark for label, but accuracy. Same marking point. Eventually, you get the same marks. Correct? So you have to do everything. So let's look at the mark scheme. What's the marks given? So again, first marking point is they check your minimum size. So by now you know how we check minimum size. Entire space, how wide is it? The widest part of your drawing must be more than half that length given there. Okay, the length of the space or the width of the space. So minimum size and lines continuous and thin. Now they said thin line. Okay. Last time they said cannot be thicker than 5 mm, but 5 mm is actually too thick already. Now they put thin line. So as long as I feel there's not thin line, I will not give mark already. Okay. So please use sharp pencil or mechanical pencil and must be HB. Draw only 4 cells and each cell touches the large xylem vessel element and at least one of the smaller cells. So you see, even though, let's say for example, in exam time, you can't focus, you can't see at all. For somehow, don't know why. Okay, and that microscope is okay. Then just simply draw lah. You learn already what is xylem vessel, right? You know the adjacent smaller cell, right? Can you draw 4 cells or not? 1 big, 3 small, and then the 3 small is touching this big one and also touching each other. You get 1 mark already. This is worst come to worst. Correct or not? You want to score as maximum as possible, you need to think how to do that. Okay? Even though you cannot see the cell, can you draw? Can. Can you get at least 1 mark? Can. Can you label the cell or not? Can. So at least 2 marks already, understand? 2 lines drawn around each cell, because every time I say got cell wall, must use 2 lines to draw the cell wall. Okay? When the cell wall are neighboring to each other, you need to draw the middle lamella. This is the middle lamella between the 2 neighboring cell wall. You must draw because can see one. Once you focus very sharply, you can see. Okay? Correct shape of cell. So I'm going to look at the shape of cell is correct. What shape? Whatever shape. Okay, you draw directly from there is correct. Label line and label to the cell wall. So cell wall point into the cell wall. I saw student label inside their drawing. Remember, all the label must be outside the drawing. What do you mean a label inside drawing? So this is the leaf, right? Very big, isn't it? You draw, okay? After that, you're supposed to draw a label vascular bundle. So the student label here and write vascular bundle here inside the drawing. So I will not give mark for the label. All label must be outside drawing. Okay, so that is very clear. You don't mix drawing and label. It's very clear. Drawing is drawing. You don't cover any of the drawing. So this one is a mathematical question. So they ask the student to, so the actual area of photomicrograph is 0.05 millimeter square. So the leaf from the figure is taken as a total surface area of 20 cm square. So what you can see in this figure is only 0.04 mm square of a leaf, where the entire leaf has got 20 cm square. And then they give you the measurement 1 cm square is 100 mm square. So what you need to do is that you need to use a figure to estimate total number of stomata in the leaf, show your working. So very easy, right? This is only mathematics. So, have I started my recording? Yes, I have. Those who come late, please make sure that you key in your attendance. So 0.04 mm square has got 4 stomata, right? So entire leaf is actually 20 cm square. 20 cm square, you have to convert to mm square. So cm square convert to mm square already given to you here. So therefore, you only need to convert times 100 mm square. Okay. We'll give you how many stomata? So it's 20 times 100 mm square divided by 0.04 mm square times 4. So that will be the answer. So how are marks given? Marks is given for your conversion correct. That means cm converted to mm correct. So this one is 1 mark. Then you divide this by 0.04, this is 1 mark, and you times by 4. So altogether 3 marks. Okay? Right, next question. One way to improve the accuracy of the estimate of total number of stomata on the leaf is to use photomicrograph with a larger area. So what we have done in the previous question is we have used this particular micrograph, which is only a small area of the leaf, this area of the leaf, to estimate the total number of stomata in this leaf. So definitely this estimation is not accurate unless the distribution of stomata in the leaf is equal. Okay, so let's say, for example, this area of the leaf you have already obtained from this part only, this part of the leaf. And you saw in this part of the leaf has got 4 stomata. So 1, 2, 3, 4. And if you only use this part to estimate the total stomata in the whole leaf, is not accurate, because the assumption that you have made here is that the distribution of stomata in the entire leaf is the same. But it may be the distribution at the bottom here, which is bigger, there are less stomata. Maybe near the margin, there are also less stomata. Near the mid-rib, there could be more stomata. So therefore, different location in the leaf may have got different density of the stomata. That means how many stomata per unit area. So therefore, based on this particular calculation to estimate total stomata here is actually not accurate. So the best thing is we need to take different, we need to count the stomata in different area of the leaf, and from there we calculate the mean, and from the mean we find out the total in the entire leaf. So that is more accurate. So that means to say that we need to have got replicates. Replicates means you don't take one reading, you take a few readings for each independent variable. Replicates are always taken for independent variable. What do you mean? For example, if you are measuring effect of temperature on the rate of reaction. Okay, so temperature you've got 10, 20, 30, and 40. You must have got 5 readings, so 15. So for each temperature, you take 3 readings. So you've got reading 1, reading 2, reading 3, and then you calculate the mean. Right? Let's say, for example, this is the height of foam in mm. So you got 3 readings and calculate the mean. So replicate means these 3 here. You got 3 readings, means you repeat experiment to get more than 1 reading. So these 3 are the replicates. So you replicate for each of the independent variable here. So now you got this leaf, only 1 leaf. So when you take the reading from different area of the same leaf, is actually considered as replicate. Okay. So the question asks the student, how to improve accuracy of your estimate? So you need to know the meaning of the word accuracy in experiment, right? Accuracy means close to the true value. True value in this case means what? The actual number of stomata in this leaf is called the true value. So how to improve? So they say one of the ways is you need to use a photomicrograph in larger area. Okay, use a photomicrograph with a larger area so that it represents a bigger area. You can see more stomata, but this is not so good. You need to say one one other way. So one other way is you need to take the reading of the total number of stomata in different areas of the leaf. So look at the mark scheme, what the mark scheme says. It says that use more field of view. Field of view is the same as take reading from a few areas of the leaf, because what do you mean by field of view? So if this is your sample, this is your leaf, right? Every time you look into microscope, you only look at a small section of the leaf, a small region of the leaf. So this is called the field of view. Whatever you can see in the microscope is called the field of view. So we say use more field of view means don't only see here, move your leaf so that you can get another field of view in the leaf, another field of view. Okay, near the margin, near the midrib, near the end, near the stem or the petiole, right? So then you get a field of view. Then you calculate the mean. So that's the meaning of field of view. Okay. So next question, they give you a micrograph, right? And they ask you, okay, they say that these two figures have the same magnification. So identify the observable differences between the leaf surface shown in figure 2.2 and figure 2.3. Record your observation in the table. So you need to write the differences and 4 marks. So first mark is given for everything that you write here must be differences. It must not be similarity. That is already the first marking point. So usually you find that 1 mark is allocated if you just answer the question. Okay, so first mark is that. Second mark, then you need to, second, third, fourth mark, you need to state the differences. First thing you can see is that, can you see the nucleus of the gut cells are very clear and nucleus of other cells also very clear? I think because they stain the nucleus, so it's dark. Whereas in this slide here,
Miss,
yes?
can you let Akilah and EJ in? They're in the waiting room.
Okay, Ken, thank you.
Thank you, Miss.
Yeah, we're in the waiting room.
So you see, here the nucleus is not obvious because they didn't use a stain, but you can see this nucleus here, right? Or maybe this is the nucleus here. Usually nucleus is this size, okay? So when the nucleus is not stained versus the nucleus is stained, then you see the differences because one is stained and one is not stained. So one is visible, one is not visible, okay? Secondly, chloroplasts. You can see here, you can see a lot of chloroplasts. You can see all this circular structure here are all they are the chloroplasts, okay? So you know that gut cells are highly packed with chloroplasts, okay? And you see epidermal cells, you cannot find chloroplasts because they have no chloroplasts. But here you can't really see the chloroplasts in the gut cell, but actually you can see a few here, 1, 2, 3 here. Other than that, you cannot see. So either you write very few or not visible. It's there. It must be there, but it's not visible. Okay. So this second thing you can talk about. Now, some of you say stomata open and close is not true because I can see the gap here. So these stomata are open, same as for this one, the stomata are open. So you cannot say open and close. They're the same. They're open. It's just that one open bigger, the other one open smaller. Okay. So what else you can talk? If you say the shape of the cell, unless you can tell me what's the difference between the shape of the cell, like the shape of this epidermal cell here and this epidermal cell is very different, but how are you going to describe? If you can describe accurately, I can give you mark. If you cannot describe accurately, like in the previous class, the student said regular and irregular shape. So the student is saying that this shape of the cell is regular. Is it really regular? That means every cell, the shape is the same, and every cell here, the shape is not the same. If you are the examiner marking this answer, will you give the mark? Or you also don't know whether to give the mark or not? Okay, the student said this cell is regular shape, epidermal cell. This cell here is not regular. I think I cannot give mark because I cannot see properly the cell of the epidermal cell. I cannot see the entire epidermal cell in this field of view. And I cannot see many of them to verify whether it's regular or not regular because I can see this cell and this cell is actually different shape. And length also different. So what do you mean by regular? I cannot see enough for me to verify it's regular versus irregular. So I cannot give mark. So if you're examiner, will you give mark? So you have to think like this when you want to choose the answer to write. So let's look at the mark scheme, the answer. There are only 3 answers here. Nuclei, okay, this is how you need to write answer. Nuclei in gut cell, no need to write nuclei again because it's redundant. A good table is table that write minimum words. Whatever you want to show stand out very clearly. So if you have written nuclei here in the future, don't write nucleus here again. Not visible, visible is obvious enough to see, okay? You can say not visible, visible, not present, present also can because you cannot see. Chloroplast, okay, visible, not visible. So no need to write chloroplast because redundant. Okay, visible and not visible. Nuclei in the leaf cell, not visible versus visible. So these are the only 3 answers they gave as the answer. In order to get the 4th mark, you need to only write features which are differences that are observable. If you write anything that's not observable there, you'll not get any marks. So it must be observable between the 2 figures and it must be differences, you get this mark. So altogether 4 marks. So have you written any other answer that's not in the mark scheme?
Sir,
Huh?
what about stomata aperture?
Stomata aperture, I give mark because one open bigger, one open smaller. So that one I will give mark.
Okay.
It's obviously this one opens bigger, that one opens smaller because since the magnification is the same, right? And also the gut cell size is different. Like this gut cell is bigger, this gut cell is smaller, and because it's same magnification, so I can say one bigger, one smaller. Okay. Another student say that, the cell wall of this cell is larger and smaller. Then I question the student, are you sure? If you say, okay, this cell wall here is narrower, but I can see the cell wall here. Can you see or not? Can you see the cell wall here or not? You need to see everything, not only see, oh, this cell wall is only 1 line. Sorry, this one is 2 lines. Then after that, you go down there and check the other slide, also same thing. The other micrograph, you see that? Oh, you see, this cell wall has got 2 lines, but just now that one also 2 lines. So it's the same thickness. So how to say one thicker than the other? But then you say, no, but teacher, this is 1 line. That one also 1 line. So your answer is arguable because I can see here and here. So your answer is half correct, half wrong. Half correct, half wrong in A level is 0 mark. You need to choose the one is 100% correct. Don't choose the one is half correct, half wrong one. You got a better choice, you choose a better choice that guarantee you to get the mark. Because every question must score full marks. Don't give an answer that half correct, half wrong. Okay? Okay, so we finished discussion already.
Miss, can I put for that one stomata arrangement? So the first one is it's facing one direction uniformly, and then the other one is irregular and random.
So let's see whether arrangement can or not. Arrangement all in different directions and arrangement all in same. Can. All the arrangement is the same orientation, right? This one, but this one is orientation is different orientation. Orientation, you see this one is like this. This one is like this. Okay, so this one is like this, all different orientation. So yes, I accept this as your answer. Very good. Any other answer you want to check?
I put here the feature as stomata state and then closed open.
So Niran says stomata, the space between the stomata is bigger or smaller can. Open and close cannot because both are open. Open and close cannot. Both are open. Okay.